Hello, everyone, and welcome back to my channel. My name is Dr. Anwar Youssef Dunbar, and this is Big Discussions 76, Science and Technology. First of all, please like this stream, please share it, and please subscribe to my channel. And that goes for you as well if you're watching this on the playback. Well, today is January 5th, 2021, and I am back with another live stream regarding the controversial COVID-19 vaccines. So I did something last week when I was out of my father's place, and I started off with a couple of articles, and then I gave a little overview of what the coronavirus is, why we care, how we got here. And then I read two articles, one of them sent to me by BGS. And then I pulled down the memoranda for the Pfizer and the Moderna vaccines. And what happened was I expended so much energy on the front end of that live stream that by the time I went through the memoranda, I think I ran out of gas a little bit and my mouth was a little dry. And so I started strong, but I didn't finish the way that I wanted to. But there was so much data in those memorandums that there's enough there for multiple videos or multiple live streams. And so I wanted to come back and talk about the safety profile, because shortly thereafter, or around that time, on Twitter, there was a woman circulating a video stating that she developed Bell's palsy from taking the vaccine. So I thought that would be worth revisiting. And also, the scientist supreme is out there. How you doing, Nicole? I'm going to talk for a little bit, and if you're hanging around, excuse me, I'll drop the link in the chat. I don't want to do a BGS all-nighter, but if folks want to talk a little bit after I get done presenting what I'm going to present, then we can do that. So, but it's about 8:30 here on the East Coast. So that's what I wanted to do. I wanted to come back and just say some things about the safety profile based upon what was in the two memorandums. I decided that I'm just going to focus on the Pfizer memorandum. For me, the data in there was a little bit easier to interpret, and their tables were a little easier to interpret. And there's quite a bit of overlap between the two memorandums. So when I discuss that, I'll just be focusing on the Pfizer memorandum. But let's jump in. So the backdrop for this vaccine. Once again, we're dealing in regulatory affairs and regulatory science. And I've mentioned this on my channel before. This is below in the description box in case you want to read about it. When you're dealing with regulatory science and regulatory affairs, what you're looking at is the federal government in this instance, and in some instances, state governments regulating and assuring the safety of products created by the public sector. And in this instance, we're looking at a vaccine created by private sector companies like Pfizer and Moderna and the other companies around the world. There's another one at the tip of my tongue, but it's not coming out. So this is in the description box below in case you want to read about what regulatory science and what regulatory affairs are. These are very, very important functions, and the general public doesn't really know how the products it uses comes to market. And those are regulatory scientists. I'm a regulatory scientist. So that's in the description box below. So let's see. So I found coincidentally two more articles, and I'm going to read these two and then we're going to jump in. But before I do, just a quick, for people watching this on the playback and anyone who's unfamiliar with what we're talking about here in terms of the virus. So everyone's probably seen these pictures. This is a cross section of the coronavirus, the novel coronavirus. This is my favorite picture. I've used this one probably the most out of all of them. I have about 5 of these pictures, but this one is my favorite. And on the outside of the capsule are the spike proteins. That's a ribbon diagram of the spike proteins. So a ribbon diagram gives you a feel for those of you unfamiliar with it, a ribbon diagram gives you a 3-dimensional structure of the conformation of a protein. In this instance, a spike protein, but it could be a receptor or an enzyme. So that's a ribbon diagram of the coronavirus's beautiful spike proteins. And the spike proteins are actually the focus of the mRNA vaccine. So these companies, Pfizer and Moderna, and the other companies, they have created an mRNA vaccine, or they're using that strategy to for their vaccines. So basically, their goal is to inject us with, as you can see in the cartoon here, encoded mRNA in the ribosomes within our cells. Will take that piece of mRNA and encode and translate the spike protein. So if you're curious about what that is, transcription and translation, I shot a video on that. But this is how the mRNA vaccine, excuse me, basically works. So they're injecting you with a piece of mRNA, and it's creating the spike protein, and the goal is for your immune system to recognize that spike protein. And so when the actual coronavirus gets inside of you, your body will be able to recognize it. X-ray crystallography. Yes, Nicole, I think that's what that is. I believe I pulled that from a paper back in the spring when VGS introduced us. So that's the backdrop for all of this. When this is what you should think about when you hear mRNA vaccine. I'm not an immunologist by training, so I did have to read up on the different immune cell types before last week's live stream, and I still would have to do more reading to tell you exactly how T lymphocytes and natural killer cells and eosinophils and neutrophils and basophils and how those guys do what they do. But you're basically priming your immune system to fight off foreign invaders, in this case, the novel coronavirus. Tiger says, good evening, chat. Okay, so I'm going to jump in and read these articles. They're short, and then we'll take a look back at the Pfizer memorandum. Specifically, I'm interested in the safety profile because that's what's in the news today. This was the first article I found. Let me enable my screen share. Let me take this picture down. Okay, there's me. That's the University of Michigan graduate school picture, by the way. That's me working on my workbench on the ubiquitination and proteasomal degradation of neuronal nitric oxide synthase. I'll probably talk about that in another live stream. But this is the article here. By the way, Nicole taught me how to do the screen share. I always credit her with that. She probably thinks I'm crazy for always saying that. So this says EU seeks up to 300 million more Pfizer-BioNTech doses. This is written by Naomi Kresge and John Folane. And this was published on January 5th, which is today, actually. Or was it yesterday? It's today. This reads, the European Union is negotiating with Pfizer Inc. and BioNTech SE on a deal that could double their supply of COVID-19 vaccine to the region as countries race to immunize enough people to tame a fresh virus resurgence. The new contract would include 100 million doses as well as an option for as many as 200 million more, according to people familiar with the talks who asked not to be identified because the negotiations are private. Governments across the EU face growing questions over the slow pace of their vaccine rollouts, and the shots from other drug makers such as AstraZeneca PLC won't be available for 4 weeks at the earliest. Okay, the attempt, what is this? Oh, tracking COVID. This is data. Total deaths report. Well, let's look at some of this. New cases reported worldwide as of January 4th, 549,954. Total deaths reported worldwide, 1,865,268. Most new cases today, 180,477 in the US. Let's see, this is some financial data. I'd probably tackle that if BGS was here. The attempt to secure more of the Pfizer-BioNTech vaccine comes less than a week after the 27-member bloc boosted its original 200 million dose order to 300 million. If the option in the new agreement were exercised, it would lift the total doses to 600 million, enough to vaccinate 2/3 of the bloc's population based on a 2-dose regimen. BioNTech's American depository receipts rose 2% before U.S. exchanges opened, and Pfizer showed little change. Member countries have pressured the EU to secure more doses as the pandemic's death toll rises. Lockdowns lengthen, and leaders worry about containing a new, more contagious strain of the virus that has emerged in the UK. While the EU has signed deals with a range of companies for access to almost 2 billion doses, only the Pfizer-BioNTech shot has thus far won approval from its regulator. A decision on a similar vaccine from U.S. biotech Moderna Inc. could come on Wednesday. However, the EU has locked up less of that shot with a contract for as many as 160 million doses. We were in talks about whether and how we can provide further vaccine doses for Europe this year. BioNTech Chief Executive Officer Uğur Şahin said in an emailed statement, no agreement has been reached and the numbers could still change. A Pfizer spokesman declined to comment. A BioNTech spokeswoman declined to comment on the number of doses involved. The European Commission is examining with BioNTech and Pfizer whether there is a way to increase supply beyond the initially contracted 300 million doses. Stefan de Keersmaker, a health policy spokesman, said Monday. He declined to comment on Tuesday about the details of the extra shots being sought. Pfizer and BioNTech are pursuing all their options to boost production capacity for the vaccine beyond the 1.3 billion doses originally planned for this year. Şahin said in an interview last month, the CEO said he was confident that would be possible, saying the partners would probably know by January or February whether and how many additional doses they would be able to offer. We're at the end here. European leaders have also come under fire for how the existing doses have been distributed in France. Administrative red tape and a lack of nursing home staff hobbled the early days of the vaccine campaign. Meanwhile, German Chancellor Angela Merkel is being criticized for tasking the EU with centralized negotiations instead of striking out on her own sooner to get more of the vaccine for Germany itself. About 266,000 people had been immunized in Germany through Monday, just over 0.3% of the population, according to the latest data from the Riki Public Health Institute, or the RKI. That compares with around 1.4% in the United States and Britain, which both began vaccinating several weeks earlier. This article is below. Let me take a look at the chat really quickly. Okay, so Nicole's here, Sheed's here. What's going on, Sheed? Menace Aptara, peace to Dr. Dunbar in the chat. Welcome. Let's see. So the second one, I'm going to take a little sip of my juice there. Let's see. The second one is, this one came to my phone a little bit earlier, and it reads, FDA warns against tinkering with Pfizer Moderna vaccine regimens after White House advisors' comments. Oh boy. This reads, this is by Shivdeep Dhaliwal. It's published today. The United States Food and Drug Administration warned against changing the dose regimen, interval, or mixing of COVID-19 vaccines in a statement issued Monday. Wow. What happened? The health agency recognized that available data indicates the use of 2 specified doses of the COVID-19 vaccines made by Pfizer and Moderna, Inc. At specified intervals. Okay, that read kind of weird. For the Pfizer vaccine, the interval is 21 days, and for the Moderna vaccine, it is 28 days. And as I'll show you, that's listed out in the memorandum that the FDA generated. FDA said using a single dose regimen or administering less than the dose previously studied in the clinical trials without understanding the nature of the depth and duration of protection that it provides is concerning. The regulator said that there is some indication that the depth of immune response is associated with the duration of the protection provided by the vaccines. Why it matters. The FDA acknowledged that there is a discussion about changing the dosing schedule or the doses so as to get more vaccines to the public quickly. Making such changes that are not supported by adequate scientific evidence may ultimately be counterproductive to public health, said the FDA. On Sunday, Operation Warp Speed Chief Advisor Monsef Saloui had suggested that Moderna's vaccine could be administered at half the regular volume to some people to further its availability. The head of the government's vaccine effort had said that discussions were on with Moderna on using the reduced dosage, but the FDA would have the final say in the matter. Okay, let's do some quick financial news. Price action. Pfizer shares closed unchanged at $36.81 on Monday and fell 0.24% in the after-hour session. On the same day, Moderna shares closed 6.95% higher at $111.73 and gained 0.31% in the after-hours sessions. Okay, so when I open up the memorandum, you'll see that the companies design their regimens so that I think the Pfizer shots, there are 2 shots that are the Pfizer shots are at 1 and 21 days, and the Moderna shots are at 1 and 28 days. And there are specific dosages that were listed out in the memoranda. And so I guess what the White House is proposing is, okay, if we give half to this person, we can give half to this person over here. The only problem is is that when you're talking about pharmacology and pharmacy, there's a thing called the effective dosage. So I think the Pfizer vaccine, according to the memoranda, each shot is, I believe, 30 micrograms, and the Moderna vaccine is 100 micrograms. So your patient might need that entire 30 micrograms to get the desired effect. So if you drop that down to 15 micrograms, well, then you might not be helping either person. So let's talk quickly about Bell's palsy, because shortly after or around the time of last week's live stream, there was a tweet going around about a woman who developed Bell's palsy. And that's actually mentioned in the memoranda that the FDA generated for both vaccines. But first, let's get a quick feel for what Bell's palsy is, because I was not very familiar with Bell's palsy until this. Both those articles are in the description box below in case you want to read those for yourself, as is this link right here. So Bell's palsy, let's read about Bell's palsy. I'm happy to see Nicole still hanging out there. Okay, so Bell's palsy. The overview. The symptoms of Bell's palsy include sudden weakness in your facial muscles. In most cases, the weakness is temporary and significantly improves over weeks. Okay, so the reason that's important is because when you're dealing with something like this, and with social media, you know, if someone tweets, "I got Bell's palsy from the vaccine," and you don't know that part right there that you can recover from it within weeks, then you may create a hysteria. So you might have a whole block of people saying, "I'm not taking the vaccine." But this is why being knowledgeable on these things is important. So I'll read that again. The symptoms of Bell's palsy include sudden weakness in your facial muscles. In most cases, the weakness is temporary and significantly improves over weeks. The weakness makes half of your face appear to droop. Your smile is one-sided, and your eye on that side resists closing. Bell's palsy, also known as acute peripheral facial palsy of unknown cause, can occur at any age. The exact cause is unknown. It's believed to be the result of swelling and inflammation of the nerve that controls the muscles on one side of your face, or it might be a reaction, or it might be a reaction that occurs after a viral infection. For most people, Bell's palsy is temporary. Symptoms usually start to improve within a few weeks, with complete recovery in about 6 months. A small number of people continue to have some Bell's palsy symptoms for life. Rarely, Bell's palsy can recur. So the symptoms, there's a little picture of, I guess, what it looks like on that guy right there. Signs and symptoms of Bell's palsy come on suddenly and may include rapid onset of mild weakness to total paralysis on one side of your face, occurring within hours to days, facial droop and difficulty making facial expressions, such as closing your eye or smiling, drooling, pain around the jaw or in or behind your ear on the affected side, increased sensitivity to sound on the affected side, headache, a loss of taste, changes in the amount of tears and saliva you produce. In rare cases, Bell's palsy can affect the nerves on both sides of your face. When to see a doctor, seek immediate medical help if you experience any type of paralysis, because you may be having a stroke. Bell's palsy is not caused by a stroke, but it can cause similar symptoms. See your doctor if you experience facial weakness or drooping to find out the underlying cause and severity of the illness. Okay, this is almost over. This is almost done. Causes, although the exact reaction to Bell's palsy, the exact reason Bell's palsy occurs isn't clear, it's often related to having a viral infection. Viruses that have been linked to Bell's palsy include viruses that cause cold sores and genital herpes, chickenpox and shingles, infectious mononucleosis, cytomegalovirus infections, respiratory illnesses, German measles, mumps, flu, hand, foot, and mouth disease. I never heard of that one before. The nerve that controls your facial muscles passes through a narrow corridor of bone on its way to your face. In Bell's palsy, that nerve becomes inflamed and swollen, usually related to a viral infection. Besides facial muscles, the nerve affects tears, saliva, taste, and a small bone in the middle of your ear. So what's interesting is, in this instance, patients and test subjects are being injected with the mRNA for the spike protein. So, it's not a complete viral particle, but as according to this, viruses are a cause of this. So maybe there's some sort of connection there with certain individuals. We're at the end here. Risk factors. Bell's palsy occurs more often in people who are pregnant, especially during the third trimester, or who are in the first week of giving birth, have an upper respiratory infection such as the flu or cold, have diabetes. Recurrent attacks of Bell's palsy are rare, but in some of these cases, there's a family history of recurrent attacks, suggesting a possible genetic predisposition to Bell's palsy. Complications. A mild case of Bell's palsy normally disappears within a month. Recovery from a more severe case involving total paralysis varies. Complications may include irreversible damage to your facial nerve, abnormal regrowth of nerve fibers. This may result in involuntary contraction of certain muscles when you're trying to move others. For example, when you smile, the eye on the affected side may close. And then finally, partial or complete blindness of the eye that won't close due to excessive dryness and scratching of the clear protective covering of the eye, which is the cornea. Okay, so for those watching live and those watching on the playback, I'm not showing this to insist that you take the vaccine. I'm not trying to convince you to take the vaccine, but I think it's just important to understand these things, because once things grow legs on social media, they can take off and cause a hysteria. And so if you don't know that Bell's palsy is something you can recover from, or that it's, in the way I understood what I just read, it can resolve for the most part pretty easily, then that adds more fear. So I think having the information is important. I'm gonna take a sip here, and I'm gonna walk through the memorandum, and then I'll throw the link in if anyone wants to come up. I don't want to talk too long. Last time I dried myself out, so. The master teacher is here. Always happy to have him come through. Okay, so the Pfizer memorandum. And this link on Bell's palsy is in the description box below this video / live stream, in case you want to read that for yourself. Okay, so this is let me make sure I had the right one here. Okay, so let me drop this one down and bring this one up. Okay, so this is a 57-page document, and I am not going to try to read through this whole thing because I think that would put you all to sleep, and we'd be here until midnight. So I don't want to do that. What's up, Dr. Goodbar? That's funny. So that's the wrong one, and this is the right one. Okay, so everyone, so going back to regulatory affairs, every time one of the federal regulatory agencies register as a product, there is usually a review and an official document generated based upon that product, so that the safety is tested, and there are everything is tested and looked at, and there's a whole panel of scientists who have to look at that, every aspect of that product, in this case a vaccine, just to check everything and to make sure it's safe for the public to use. So that's what this is. And I imagine that with the scrutiny that this vaccine is under, I imagine all these people had quite a few sleepless nights. You could see the team here who worked on this. There's at least 20 scientists there who worked on this. Okay. So I'm gonna kind of, yeah, that Nicole says that PDF is dense, Dr. Dunbar. Here, I'll put it up here. Oops, almost spilled my juice. That PDF is dense, Dr. Dunbar. I need some time, LOL. This is below if you guys want to take a look. Nicole, I'm sure you're curious. It's down there if you want to take a look for yourself, and it's also available online. So this isn't top secret. They released this. Okay, so let's just jump through. Here's a table of contents. If you decide to read this for yourself, there's a glossary at the front. When I go through the safety section, adverse event, that acronym and that term will come up. Also, serious adverse event will come up as well. But this is in the front of the document in case you want to read that for yourself. The executive summary, this is kind of like the abstract, summarizing the document. That's the introduction that gives you a nice background on COVID. Let's see. Safety data. Okay, so as there have been a lot of speculation and a lot of some conspiracy theories about what is actually in the vaccine. I've heard they're putting microchips inside of us. I've heard that they're putting nanotubes inside of us. I've heard all kinds of things. And as a pharmacologist / toxicologist, one of my first questions was, what's in this thing? So if you look in the memorandum on page 11, it tells you right here, vaccine composition and dosing regimen. So I'm going to read this paragraph. The Pfizer-BioNTech COVID-19 vaccine is a white to off-white sterile, preservative-free frozen suspension for intramuscular injection. The vaccine contains a nucleoside-modified messenger RNA encoding the viral spike glycoprotein of SARS-CoV-2. The vaccine also includes the following ingredients: lipids. These lipids, I'm not going to read all of those out. Potassium chloride, monobasic potassium phosphate, sodium chloride, dibasic sodium phosphate, dihydrate, and sucrose. Okay. The Pfizer-BioNTech COVID-19 vaccine is supplied as frozen. The Pfizer-BioNTech COVID-19 vaccine is supplied as a frozen multi-dose vial. So there are 5 doses in each vial. The vaccine must be thawed and diluted in its original vial with 1.8 milliliters of sterile 0.9% sodium chloride injection, USP, prior to administration. After dilution, the vial contains 5 doses of 0.3 mLs per dose. After dilution, the multiple dose vials must be stored between 2 degrees C to 25 degrees C and used within 6 hours from the time of dilution. Okay, the Pfizer-BioNTech COVID vaccine. So this one is 30 micrograms. So each shot is 30 micrograms. It is administered intramuscularly as a series of 2 30 microgram doses. So if you cut this in half, that's 15 micrograms per patient. And again, most pharmaceuticals, you have to achieve a certain dosage for it to show efficacy, meaning in order for it to work. Okay. So let's see. I'm going to skip down here, and all this is below if you want to read it. Let's see. I talked about the efficacy last time, so I'm not going to spend a lot of time on that. But this is basically their study design for the trial. So dose 1 and dose 2 are 21 days apart. So that's the vaccination period, and they had a follow-up period going out to 2 years. So these two studies, the Pfizer and the Moderna studies, they're still ongoing, and they state that right here in the memorandum. So, for all the people dosed, the vaccine dose and the placebo, they basically evaluated them after their dosing for efficacy and also for safety and adverse reactions. And this is going to go out for 2 years. We'll look at the chat for a second. Okay, we got a piece to Nicole Ali out there. Nicole is reading it right now. Okay. Let's see. So they list out basically the populations, their sample size, and who all was in their samples, their sample group, why certain people were discarded, why some people didn't complete the study. All that's described. I'm not going to spend a lot of time on that. Let's see. Disposition of all randomized patients, phases 2 and 3 of the safety population. So this is a breakdown of the safety population. And as you can see, the vaccine treated group had 18,904 people in it, and the placebo group had 18,892 individuals in it. So they wanted to, numerically create 2 equal-sized group, probably most likely for statistical analysis on the back end, because that's an important part of a study like this. And this breaks down their sample size or their sample groups by age and sex and race and those things. Let's see, demographics and other baseline characteristics. So this is further characterizing their sample set and the patients tested. Okay. So this whole section here deals with efficacy. So it's dealing with how well the vaccine actually worked within that time tested. I'm not gonna spend time on all these tables. Instead, I'm going to focus us on this figure here. This is shrink that down a little bit. This is entitled Cumulative Incidence Curves for the First COVID-19 Occurrence After Dose 1, Dose 1 All Available Efficacy Population. I'm going to read this paragraph because this is describing that figure. Based on the cumulative incidence curve for all available efficacy population after dose 1, COVID-19 disease onset appears to occur similarly for both the vaccine treated and the placebo groups until approximately 14 days after dose 1, at which time the curves diverge, with more cases accumulating in the placebo group than in the vaccine treated group. And there does not appear to be evidence of waning protection during the follow-up time of approximately 2 months following the second dose that is being evaluated at this point in time. Okay, so much of the data in terms of the the efficacy section shows that there is protection from the infection with the vaccine. And I talked about that last week. So I'm going to go down to the safety section, and then I'm going to stop and drop the link in the chat. So in terms of safety, so you know the thing works, but then the question is, going back to the Bell's palsy, is it safe? You know, what are the side effects? What are the potential side effects? And this is the thing I was thinking about when BGS was talking about it, and whenever, you know, people were talking about, am I going to take it? How safe is it? They're putting chips in us. They're putting nanotubes. My thought was always about the safety. So this paragraph reads Table 14, and Table 14 says study C4591001 safety overview, ages 16 years and older. So in this Pfizer study, the ages ranged from 60, I believe, to 60 to like 70 or something. But they didn't test teens or children below 16 years of age. Table 14 below represents an overview of all adverse events in the phase 2-3 safety population. A higher proportion of vaccine recipients reported adverse events compared with placebo recipients, and this imbalance was driven by reactogenicity, or solicited adverse events, reported in the 7 days following vaccination, and unsolicited adverse events corresponding to reactogenicity symptoms among patients not in the reactogenicity subset. Proportions of participants with serious adverse events, deaths, and withdrawals due to adverse events were balanced between treatment groups. Okay. So let's see. So if you look at Table 14, participants experiencing at least one immediate unsolicited adverse event within 30 minutes after vaccination. Dose 1. So the percentages are in the parentheses. And so for immediate unsolicited adverse events within 30 minutes after vaccination, there was between dose 1 and dose 2, there's basically no difference. So the solicited injection site reaction within 7 days, there was a difference there, 12.8% in the placebo and 78.6% for the virus, for the vaccine. And then for dose 2, there's 10.6%, and then there's 73.1% for the vaccine treated. So there appears to be something happening at the injection site there. Let's see, solicited systemic adverse events within 7 days. For dose 1, there was 47% in the placebo and 59% in the vaccine treated. And at dose 2, 33.8% versus 69.9% for the vaccine treated. So there's a slight difference there. Let's see, from dose 1 through 1 month after dose 2, unsolicited non-serious adverse events, there's a slight difference between the placebo and the vaccine treated, 12.5% versus 27%. And then severe adverse events, there's no change. I'm gonna take a quick look at the chat. It's one thing about the immediate effect. How long does the mRNA keep replicating? That's a good question. We might need the scientist supreme for that one. But give me a few more minutes. Okay, I'm going to jump to the next table. Okay, I thought this was interesting. Table 15, frequency of solicited local reactions within 7 days after each vaccination, reactogenicity subset of the phase 2/3 safety population, 18 to 55 years of age. So you have 2 doses. Dose 1 is these first 2 columns. Dose 2 is the second 2 columns. So they were looking at pain, redness, swelling. And these are local reactions. So I'm interpreting that as being at the injection site. Let's see. I'll just read this characterization. For each age group in the reactogenicity subset and overall, the median onset of local reactions in the vaccine group was 0. They had vaccination 0 to 2 days after either dose and lasted a median duration of 1 to 2 days. For both age groups, injection site pain was the most frequent solicited local adverse reaction. After dose 2, the younger age group reported any pain more frequently than the older age group, 78 versus 66%, and pain characterized as moderate, 27 versus 18%. A similar pattern was observed after dose 1. Injection site redness and swelling after each dose were generally similar for both age groups. Okay. So pain, so any pain. So for the first dose, any pain, yeah, there was more pain for the vaccine injected, and the same was true for the second dose, and that's a significant difference: 14% versus 83%, and 12% versus 78. But there were times when I got the flu vaccine, and my arm was sore after getting the flu vaccine. So that's not unusual to me that something like that would happen. And they further break these down: mild, moderate, severe. There were no severes for pain in terms of redness. There was for the first dose, there was a small difference between the placebo and the vaccine treated. For the second dose, there was a 0.7. I don't see any stars here, so I don't know if that's statistically significant, but there's a change between the placebo and the vaccine treated. And I don't really see a big difference for severe redness. Swelling, yeah, you have a 10-fold increase for any, and then for dose 1 and for dose 2, you also have a greater than 10-fold increase. I'm going to do 1 or 2 more tables, and then I'll stop. Let's see. So this is frequency of solicited local reactions within 7 days after each vaccination, reactogenicity subset of the phase 2 and 3 safety population for individuals greater than 55 years of age and older. So BGS, this is your group. So this is similar to the previous. What's the age group here? This is 18 to 55. This is 55 years and up. So it looks similar in terms of the difference. So for pain, you have a big difference there between the placebo and the vaccine treated for the first dose, and the same is true for the second dose. For redness, you have a minor change there. For any, there's really no significant change for the severe. For the redness, you have a 10-fold change here for the placebo versus the vaccine treated. And then for swelling, there's a small change, 1.2% to 6.5%. I'm looking at the severes. This is the severes for me as a toxicologist, as a pharmacologist, toxicologist. Severe really rings a bell for me, and I'm not really seeing any of that. So the next set is solicited systemic adverse events. So I'm going to read this characterization. For each age group in their reactogenicity subset, younger, 18 to 55 years of age, and older, greater than 55 years of age, and overall 18 years and older, the median onset of systemic adverse events in the vaccine group in general was 1 to 2 days after either dose and lasted a median duration of 1 day. The frequency and severity of systemic adverse events were higher in younger than older age groups. Within each age group, the frequency and severity of adverse events was higher after dose 2 than dose 1, except for vomiting and diarrhea, which was generally similar regardless of dose. For both age groups, fatigue, headache, and new/worsened muscle pain were most common. Okay. So table 17, frequency of solicited systemic adverse events within 7 days after each vaccination, reactogenicity subset of the phase 2/3 safety population. Excuse me. 18 to 55 years of age. So this first set is looking at temperature. I recall there was an article of a nurse that I presented last week, and she said that she did witness changes in temperature. She said she felt her temperature. She developed a fever. I don't recall if that was the first dose or the second dose, but it's in the description box for last week's live stream. So here they were looking at differences, same setup, 2 doses, placebo versus vaccine treated in both instances. And so keep in mind here, they're monitoring fever, and they listed out 38 degrees to 40 degrees. Normal human body temperature is 37 degrees. So some of the patients after the first dose experienced an increased temperature greater than or equal to 38 degrees Celsius. And there was, this was more profound after the second dose here. Let's see, 38 degrees to 38.4 degrees. There was a small difference for the first dose. Once again, it seemed to be more pronounced in the second dose. So you're looking at 0.2% versus 9.2%. Let's see. There's a small difference here for 38.4 to 38.9. Not much to see there for dose 1. For dose 2, it seems that dose 2 is more was more likely to cause the increased temperature. And there wasn't much going on for greater than 40 degrees. There wasn't anything going on there. Okay. So they looked at all these different things here: fatigue, headache, chills, vomiting, diarrhea, new or worsened muscle pain, and new or worsened joint pain. So I think I'll present this table and then I'll stop there. I wanted to give everyone a feel. There's so much news going on, but the FDA actually looked at this, and this is the data that they looked at to green light the thing. So for fatigue, for dose 1, there's a difference there for any 33.4% in the placebo. That's interesting that there is some effect in the placebo, with 33.4% in the placebo, 47.4% in the vaccine treated. And then if you look at the second shot, you have 22.8% in the placebo versus 59.4% in the vaccine treated. So for whatever reason, the second dose seems to generate more of these effects, which is interesting. Let's see. So for mild had a slightly higher increase in fatigue for dose 1 versus dose 2. There wasn't a big change for the moderate group, but there was a significant change for the moderate group following the second dose. And for the severe, well, this is almost threefold higher, but there's a small difference there. So headache, normally when you get a fever, you may get a headache as well. So there's a significant difference there for the placebo treated versus the vaccine treated for dose 1. And there's a difference between the placebo treated and the vaccine treated for dose 2 for any. So I want to be careful about saying significant, because that Nicole will vouch for this. That means, in the science world, that means statistically significant. And I don't see all of our standard tests here. So when I say significant, I was just talking about in terms of magnitude, in terms of change. So these values are doubling. So chills, there's an effect on chills for any at dose 1. But once again, the second dose is giving a much more robust effect. So there's something about the second dose. And let's see. I think everything else here is pretty much, it's pretty much, So I'm gonna stop there. Nicole, if you want to come up and say a few words, I'm going to drop the link in the chat. I'd be curious to hear what you have to say about some of this. Scientist Supreme and BGS, if you want to come in as well. Let's see, it's 9:32. I just wanted to give everyone a feel for. I just want to give everyone a feel for some of this stuff from a regulatory standpoint, because there's so much news flying around, and there's so much conspiracy on Twitter, and I just wanted to give everyone a feel about what I saw as a regulatory scientist when I looked at this information. So this I see Nicole said in the chat that she'll probably break down pages 11 to 19. This is a 57-page document. So there's a lot in here. So this would, to hold the attention of an audience, through all of this would be a gargantuan task. And I haven't sat down and read over everything with a fine-tooth comb myself. So I just wanted to give everyone a feel for what's going on. So BGS has entered the building. Hello, sir.
Hey, doctor. What's going on? Dr. Goodbar, what's up? Pretty good.
I was listening to your live stream from yesterday, last night, this morning. So
okay. Yeah, it's supposed to be an hour and it turned into 4, but you know how that goes. Is this amount of information that they come forward with, is this normal, or does it take longer than this?
Well, they had to fast track it.
Yeah.
Someone said that, I read another article that said that a lot of these pieces were already available in terms of basic research, and some of this they had to kind of just stitch together. But in terms of speed, this is much faster than typical vaccines. But in terms of the amount of information, I've never registered a vaccine myself, so I don't know. Okay. But I imagine the need for transparency for something like this was very paramount.
Yeah. My thing is that since it's a fairly new process, my thing is more about the long-term effects. Like like Nicole says, it doesn't have it's not terminating. So just depending on the body's ability to actually shed the particles and get rid of the virus, so it doesn't have any long-term effects. I'm just wondering, will this say once you put it in, does it keep replicating, or does the body control it, or is there an end to it where it's not effective anymore? I'm just curious. Well, nobody knows. So,
we have to take the flu shot every year. So I see Nicole's here. Perhaps she could weigh in on this. But you need to take 2 of them. Well, they designed it to take 2 of them. So I imagine, yes, it will fall off after a certain time. And I remember someone asking that question. We have to take this several times a year. I don't know. What's going on, Nicole?
Hey, what's up? What's going on with y'all?
Here's our expert, man, because we're waiting for the word, for the ism.
New Year? Question? That's all I can say.
That's about it, huh?
New Year 2021.
Yeah. Oh yeah. It's interesting. Okay.
Yeah, I don't know. Like I've been that stream earlier like wore me out, but just talking to my colleagues and stuff, like I'm coming from a different perspective. That's what I want people in the chat to understand. You know, Dr. Dunbar, much respect to you. You're coming from a regulatory perspective, also toxicology and pharmacology. So you're looking at things a certain way, and I'm looking at things a different way. And what BGS, what you were asking in the chat, like that is a good question because I've never worked with the mRNA, the nucleoside mRNA vaccine, you know, at all, ever. Like nobody's ever done that. Anybody I know, we've never done it. We've only done a transfection, and it's not in that order. Like we don't use that much fat, you know, that many lipids as a carrier molecule or as a preservative. Usually fats are used as a carrier molecule, but the sodium chloride, potassium chloride, that type of stuff is used as a preservative and stabilizer. But we don't we don't use, and we definitely don't use that much. You know, this is 30 times the amount that I would use for standard transfection.
Wow. Okay.
Because we use 1 microgram or 1 micromolar, depending on how you made your virus, how you amplified the virus, amplified the DNA that you want to use. First of all, we use DNA. We don't use RNA because it's unstable. So when we do our transformation, we put the plasmid. We need to put that into some the competent bacteria, you know, DH5 alpha type of bacteria, E. coli. You grow them up in the laboratory, you know, and everything. If they live for 18 hours and they grow, then you take those bacteria, you grow them again, and you know, you add your antibiotics. So whatever bacteria in the air, it's not going to survive because the antibiotic is there, right? You take that slurry of bacteria, spin it down into a pellet, you lyse the bacteria, you take out your DNA. It ends up being like water in a tube. And Don Calypso knows this whole process, but you run your MIDI kit and you get your DNA, you quantitate the DNA, you only transfect 1 microgram. I mean, we can go down to like 100 nanogram. Maximum I've ever done is 5 microgram because the cells were very resistant. But I ain't never put no 30 micrograms of anything into a cell or a mouse. You know, that's ridiculous. It would kill them.
So Nicole,
you know, so this is different because this. Sorry, go ahead.
No, I'm sorry. I cut you off. I'll wait to get done.
No, I'm just saying this is different because humans are obviously bigger and then have a more complex immune system. And that's also has to do, I said on Brother X's channel, Christmas Eve, it's also has a lot to do with where they inject you. They inject you, it's intramuscular. So there's a difference between IV injection, which is what I would do, injecting the tail of the mouse. Like if I want to introduce a virus, I inject in the tail of the mouse, or I do intraorbital injections, inject into the eye. Sometimes they live, sometimes they don't. You know, sometimes I mess up. You know, okay, you know, but if I don't want to do intraorbital, then I can do if I want a systemic reaction that's very fast, I'll just open them up and inject in the heart.
Okay.
You know, immediate. But this is in the muscle, so the uptake is very fast in human beings because we use our muscles for pretty much everything, right?
Mm-hmm.
And the uptake is fast. I mean, you see people having responses, like my dad said, the VA, like in less than 20 minutes, people have responses.
Wow.
So that's
Okay.
expected. But
Mm-hmm.
what's not expected is the types of symptoms and the overwhelming amount of not knowing what is going to happen, like how long, like you said, how
Mm-hmm.
long it's going to be in the body and all that kind of stuff.
Yeah. Well, yeah. Because of the emergency that we're under, the gun that we're under, they don't have time for a long-term study. But I
Right.
was just curious. You know.
Yeah, it's still ongoing. Those two they had, one was multiple countries, United
Mm-hmm.
States, Turkey. Um, I think China might have been on there too, but
Mm-hmm.
China has their own. They have Sinovac. China has its own going on, but there was like what 6 or 7 countries on one of the tables. That was one that's ongoing,
Mm-hmm.
and the other one was Germany by itself, ongoing.
Mm-hmm.
Russia
Yeah.
had one. A lot of people, a lot of countries had their own thing going on. It's not just Pfizer, AstraZeneca, Moderna.
Yeah. Yeah.
So
Right
that's
now,
what keep
Cherry
in mind.
said that Oxford's version is DNA. Is there
Yeah.
is there a so said a basic difference between using one versus the other, or is
Mm.
there an advantage or a disadvantage one versus the other?
Yeah, there is, because the mRNA is going to be faster but more
Mm-hmm.
robust
Mm-hmm.
effect, and the DNA takes longer. You have extra step. You got transcription, and then you have mRNA processing. Then you have the translation
Mm-hmm.
that needs to occur. Also, introduction of a DNA vaccine may be different, meaning like where they inject you,
Okay.
it may be different. So they may not have to use as much, because they know it's going to take like 5 days for you to have some kind of response or whatever. But or they could inject more if it's DNA, depending on what it is they're doing. What is the like what are the carrier molecules they're using? Like some of
Mm-hmm.
those lipids these are not small lipids. They're very big, and they're using them for a certain reason. I don't know, like what the composition, or not the composition, but what the how much, like what is the concentration or dosage of each lipid that they're using per microgram of
Mm-hmm.
RNA. I don't know, or per microliter, because they're giving you 30 micrograms in 300 microliters, and that's a diluted form.
Mm-hmm.
It's diluted what 5 times or something. So it's actually 150 micrograms, right? I think in a vial. You have to dilute that down 1 to 5 or something, or 1 to 4 or whatever. You have to dilute that down. So those vials they have very high concentration per vial.
Mm.
And
Yeah.
each vial is supposed to be like for so many people or whatever. But that's a lot. Like 300 microliters is not a little bit to put in your body. It may seem like it is, but it's not. Like it's a lot, And 30, like I said, 30 micrograms, that's like, because the idea is that every microgram of that solution is going to do something,
Mm-hmm. It's gonna
you know.
replicate itself.
So Nicole,
Yeah.
well, first of all, I want to thank everyone for the cash apps: Aquateki, Ramon, Palacios, Nicole, and Reynald Cherry. Thank you, Nicole. As a pharmacologist, because we're very concerned about what the actual molecule does and what the formulations do once they get in the system. You sounded concerned about the lipids.
Mhm.
Why are you concerned about the lipids? Because normally lipids are pretty benign, right?
Well, because if it's a carrier molecule, if they've I don't know how they formulated it, because you can have proteolipids, you could have vesicles. Vesicles are made of lipids, vesicles or exosomes. Um,
There
you
you
know,
go.
you know what I'm saying? Vesicles or exosomes or
Uh-huh.
oncosomes.
Uh-huh.
It's a lipid taxi cab basically, and it's a micelle. So it's lipid layered, single layered, some of them bilayered. They're very, complex. And inside is a little piece of DNA or a little piece of RNA or a little piece of protein, traveling around from cell to cell. They pass the notes back and forth basically.
Mm-hmm.
And So I think about that, like, what is the formulation? How much lipid is there? Because you inject this into the muscle. It's not this is not like they took lipids from a cow and like something natural. This is a unnatural synthetic formulation of lipid that they
Okay.
have, that they're not just they're not just saying, oh well, we got some lipid, we got some vegetable oil, we injected into you. No,
Mm,
that's not what that is. So I worry about that. And then I also think, like, it's, they want it to be speedy. They want a very, like I said, robust response. And that's what they're getting. I have never seen somebody get an injection and like 15 minutes later they have this, Supertriz probably has seen that happen.
mm,
But he said it was based on vasodilation, which is
mm-hmm.
different than what I'm talking about, which is more of the biochemical aspect of it, of the vaccine itself. But with vaccines, normally, think about when you got a vaccine. I had to have chickenpox vaccine twice because I never got it. And when I got it both times, it took like 5 days to see like a little pimple. Like I did, I never got full blown chickenpox ever. So, for me, thinking about that and then thinking about the standard that everybody
Mm-hmm.
knows, like 3 to 5 days, 3 to 5 days. And
Mm-hmm.
you're using people who are healthy, relatively healthy, right?
Mm-hmm.
16 or older, 16 or older, or whatever it is. So that concerns me. That just may be my occupational hazard coming in, because I'm studying lipids anyway. But but yeah, I just think about that. Is why you got so many lipids? Like, why you need
Yeah.
all of that? It just, that concerns me when I think about it.
Well, let's take
Yeah.
a. Hey, I see sanity question. I see you asked a question. I'm going to get to that in a second. But let's take a quick look at the what's in the Moderna
Okay.
vaccine to compare, Nicole,
Oh, okay.
because I have that locked and loaded. So where is it?
It's a good thing we have you guys, man.
The Moderna COVID-19 vaccine is a white to off-white sterile preservative-free frozen suspension for intramuscular injection. The vaccine contains a synthetic messenger ribonucleic acid encoding the pre-fusion stabilized spike glycoprotein of the SARS-CoV-2 virus. The vaccine also contains the following ingredients: lipids,
Mhm.
and I don't recognize all of those, but cholesterol and 1,2 diasterol, S-N-glycerol, 3 phosphocholene. I think that might have been all one lipid. Tromethamine,
Mhm.
hydrochloride, acetic acid, sodium acetate, and sucrose.
Mhm.
And I believe it doesn't have to be frozen. I don't think it has to be frozen like the Pfizer. But
Yeah, so it's just like how it's formulated, like what you add first, second, third, whatever. Like, some of them don't have to be. But see, it's something you said in there. You said fusion. So that could have something to do with this. So if the active ingredient, which is the mRNA itself, the other one was a nucleoside. This one is not a nucleoside, I don't think. So Moderna did something a little different than Pfizer. They made it so it could be transported without being hit minus 90 degrees, not minus
Mm,
80 degrees, which is crazy.
mm-hmm.
Because if you want to take this to Africa or
Yeah, that's
any
true.
hot environment, how are you really going to keep that cold and transport it long distances? And you have to make sure it's cold. I mean, it basically has to be on dry ice and stuff, or liquid nitrogen, even if you really wanted to be safe about it, and make sure that it comes to a certain temperature. But this is a fusion type of vaccine, so it's like, okay, y'all doing too much. Y'all is doing, and I understand what they're saying. They're saying that the they basically have fused the sequences of or determined the sequences of the spike protein, different components of the spike protein, so that your body will think that you've been infected with all of that, all, because that's all fused together, right? And so when you get the vaccine, your body gives off the immune response not only to like one part of the spike protein, but the whole thing. So that makes sense. That's more comprehensive. but that word fusion just kind of makes me a little nervous. I don't know. It's like certain words throw me off, and it's not me being a conspiracy theorist. It's me, being a scientist. Because when we use
Mm-hmm.
fusion, when we use the word fusion, it's something that's bad. It's associated with
Oh,
something
really?
bad.
Okay.
Yeah, we put like temperance erg fusion gene is something you only see, well, primarily you see in cancer patients, like very bad cancer patients that are just jacked up genetically.
Mm-hmm.
Any kind of fusion, like ALK fusions, certain signaling protein, extracellular protein that have fused together.
Mm-hmm.
You dealing with some issues right there, because that means that it's gonna make it harder for us to treat that person. It's gonna make it harder for us to find out, like they may be more likely to mutate, those tumor cells if they have fusion proteins. Because a lot of
Mm-hmm.
times fusion proteins will compensate for each other, and that's why I worry. But this could be totally different. They're talking about a vaccine, so it could be different. But the fact that that is in the title or in the description of it, it's like, I don't know.
Okay. Oh, Winston has a question here. How long is usually given to test a vaccine? And I pulled a picture from one of our earlier live streams here, and that's not a very good resolution. But I'll just say it could take years, Winston.
Oh years. Yeah.
So they fast tracked this one.
Yeah, I think fast track is the is understatement, man. Like warp speed.
Yeah, I mean they did name it warp speed. I was like, y'all.
Yeah.
What are your friends doing in DC, Dr. Dunbar? You should know. What the heck? What is y'all doing? Like, come on.
Hey.
I know, Dr.
We got to get the economy rolling again.
We got to get it rolling again. Yeah, that's what I said that back in November, and that's
right.
Because what's his name? Jerome Powell actually tipped it off when he said that the vaccine's not going to be a panacea. You're going to need some kind of stimulus. I said, okay. I said, this is going to happen. They're going to try to punch this through. They're just going to see how many restrictions they're going to make to actually encourage, for lack of a better term, people to actually take this vaccine.
Yeah, and I worry about older people, and I worry about people with pre-existing conditions. That's just saying Americans, but I just worry about like people with severe immunocompromised patients, pregnant women and whatnot, even children. That's what I worry about. And then everybody pretty much knows somebody by now that has it. Like, it's like what, 1 or 2 degrees of separation. Like, somebody, you may know somebody, or you may know somebody who knows somebody. You know what I'm saying? Like, it's real close. For me, I know somebody and a few people, and it's like, okay, coworkers, friends, like it's crazy.
Yeah, we got congressmen dropping dead. So
yeah,
is that right?
Yeah.
One from Louisiana. He died 2 weeks. Well, actually, a week ago. He had it for 2 weeks, and we got one. I was asking Nicole about. There was one congressman that from Pennsylvania that had an aneurysm, but he tested positive for COVID-19. So they're trying to figure out if the aneurysm, if an aneurysm could be a byproduct of COVID-19. That's been proven. So we'll have to see. And then we're looking at looking at Dr. Dre, who just had an aneurysm.
Yeah, that just happened today, and
oh wow.
I already know, like, it's crazy down there. Like, I already know, like, it's crazy because I looked at that when you told me. I was like, the videos are like 24 minutes, 15 minutes. I'm like, whoa, okay, this literally just happened like this afternoon or something, this evening. That it does affect your brain. They know it affects cognitive abilities and obviously the circulatory system. I don't know if he was positive, but that would definitely that could definitely lead to, they said, aneurysm or stroke.
Yeah.
He's not a young person.
When the best treatments are anti-malarial drugs, that means this little bugger is different.
Yeah.
So there's something to this one. And, it takes a genius like you, Nicole, to actually decipher it.
I'm like, this is too much for me. Like, I've been trying to deal with other stuff that's like totally not on this, looking at reading a lot with like on metabolism and
Mm-hmm.
like trying to focus on my own work, and I have a lot going on, but
Yeah.
this right here, like these vaccines, like we're gonna have to take it, if you want to travel. I want to
Mm-hmm.
travel soon, I have to, figure out which one is best. People are already signing up for it.
Oh,
They
really?
got waiting lists in different places for it. And I know the governor in California was talking about, rolling out all these hundreds of thousands of vaccines. But my issue is he wants to introduce so many hundreds of thousands of Pfizer and so many hundreds of thousands of Moderna
Yeah.
vaccine. I was like, wait a minute, like they work differently.
Yeah, but I really don't think it's about which one is the best. I think the thing is, can you get them a vaccine and can you get these people
Back to work.
certified and go back to work? And the biggest thing that I've understood is that Donald Trump didn't order enough of the Pfizer, so a lot of the Pfizer drug went to Europe, and even in Europe, there's a shortage.
Mm-hmm.
So a lot of a lot of the vaccines went to Europe already, and so there's a shortage. So they had to, bring in a second, and I think they're trying to bring in a third, if I'm not mistaken. Do you hear about that, Dr. Dunbar?
I did not.
Okay, I think they're trying to bring in a third. I think they was trying to bring in a third, but that one was rejected for whatever reason. But I think they're trying to bring in a third just to get enough doses of whatever vaccine to actually certify people. That's the way I'm looking at it. Now, whether it works, because I think there was a nurse that said she took it, and then a couple of weeks later she tested positive. But like Nicole
Yeah.
probably would say, it could be that the vaccine caused a false positive in her system.
Mm-hmm.
So there's
That's
a lot,
true. Or it could be that the vaccine does not cover, you have to think about when they put the formulation of the vaccine together.
Yeah.
And they were working on it. Like these trials are ongoing, and they're working on it
Mm-hmm.
during a time where there weren't that many mutations in this country. A lot of mutations were in India, China, places like that. And now that is out, the vaccine is out to be used and readily available. That's not going to stop the mutations from happening. So that's
No.
also a possibility too.
Yeah, they got
You got the vaccine,
Yeah.
but you got infected with a mutation. So, and that vaccine doesn't cover it. That's the thing I was worried about, because when
mhm.
you talk about efficacy, you're talking about not just the graph that you showed table. I think no, it was figure 2 you showed in that document, not just figure 2 where you drop infection rate, but also the like the recovery and everything, but coverage, recovery of the patient, meaning that they have, they may have symptoms, but the symptoms subside over time. But coverage, like the sequence coverage of the vaccine, that's proprietary information, obviously. Because if we knew what that was, I don't think people would be running to get the vaccine, Right there. The red line is the placebo, the blue line is it dropped.
Wow.
And it took
Okay.
2 weeks to do that. So within that 2 weeks, but I mean, and that's the other thing, like you don't even know, like you're giving people the vaccine and then okay, you can go home. Well, you think people just gonna go be like, oh, I'm gonna go straight home, straight away? No. They gonna go to Kroger and Walmart and holler at their
Yeah.
friends and whatever and
Mhm.
get on Facebook Live or whatever whatever they're doing, and then they're gonna go home. Like unless there was a stringent protocol or you
Mhm.
had cameras on these people and made sure they didn't go anywhere except go home, come back and get your blood drawn. Go home. Like unless you did that, how do you know if this person got infected while they were vaccinated? That's my
That's true.
my issue with it. And you got infected with a mutation and didn't know, because with the mutation, with so many mutations now, you are asymptomatic. Like a
Mhm.
lot of people are asymptomatic right now,
Mhm.
just walking around chilling with no
Yeah.
mask on.
Yeah.
Like
Yeah.
some
Yeah.
fools out here in the street.
Yeah. Yeah. Because just recently the two main mutations is the one in coming from the UK, obviously, and also there's a new one in South Africa. So
Yeah.
So Nicole,
Yeah,
so when you said,
I knew that would happen. Yeah, go ahead.
so Nicole, when you said coverage, are you referring to the sequences on the spike protein?
Yeah, because you see how the green one at the top, like that's the
Mhm.
one probably that's gonna be facing outward
Mm-hmm.
from the like you have to imagine this image multiplied like hundreds of times
Mm-hmm.
on the outside of the I call it the buckyball, but the capsid, the ball itself, the
Yeah.
coronavirus ball itself. So if that green spike protein portion, if 50% of the green parts just change its shape just a little bit, that's a mutation.
Wow. Okay.
You know,
Wow.
it may not be a large mutation, like it may not be dramatic,
Mm-hmm.
but it may be a point mutation or maybe a missense mutation that just mutation that just don't make any sense. Where maybe 50% of the parts of the spike protein are not functional.
Yeah.
Mm-hmm.
So you would think, oh well, if 50% of the spike protein particles are not functional, then the virus should slow down. Not necessarily, because all the viral particles in your body are not going to have that mutation. You know, the red part might mutate on some of the particles, 50% of them, making it more aggressive. And the green parts, maybe all the green parts go dormant. Okay. So that makes a difference. That's a mutation. That's a double mutation right there that you have. You know, you might have a frame shift mutation where the protein is not aligned properly. You know, anything could happen as far as mutations. It's random.
Mm-hmm.
And
Yeah.
then if you get the vaccine, but then the green part is on this image, if that green particle of the spike protein is not there at
Right.
all,
So it all
and that's what
depends
you
on
get.
what.
That's what you get infected with, and you end up like, I got vaccinated, but I'm positive. What's going on? Yeah, that's the problem.
Yeah. So it all depends on which epitope the antibody
Yes,
is recognizing.
yes. You trying to use all the technical terms? You know what?
I'm trying to get.
I'm trying to keep it real. Yeah, the epitopes and everything, and the and then with the antibodies, you have your FC change and things like that. But the different epitopes on this protein that are needed to signal, they have a specific job. The part that's gray may not be. It may be gray because it's not as significant. The parts that are in color are like
Yeah.
really important.
Okay.
That's usually how they do the images. This is a lot of alpha helices and like some beta sheets on the blue part.
Yeah.
A lot of beta sheets going on. Alpha helices are very they just change shape,
Mm-hmm.
they're very flimsy, unless they're like um unless they're like stabilized with the beta sheets or something. They're kind of flimsy and they just kind of hanging out there,
Mm-hmm.
until our virus is active. Then it's like, okay, I have a job to do. But mutation, it can make it real flimsy and just be flopping all over the place. It has like no
Yeah.
function.
Okay.
Yeah.
So that's another thing. And that does not mean if parts of the spike protein go non-functional, that doesn't mean you're not gonna have an immune response. You still might have a cytokine storm.
That's
Why?
true.
Because this is there and it's not supposed to be there.
Yeah.
You
Yeah.
know what I'm saying? So don't get your vaccine and then be like, I'm going drinking or something. Like, no, you need to go home.
You had to go home. Well, okay.
Go home, go to bed. Like, just get in the bed. Don't pass go. Just go straight home and get in the shower and get in the bed. Like, that's your best thing. Like, even when you have children
Mm-hmm.
that get vaccines, little babies and little kids,
Mm-hmm.
they tell you not to kiss the baby in the mouth, not to, let people around the baby unless they're in the household, and don't take the baby out too much. Keep them home, keep them warm, make sure they're hydrated, things like that. They've been saying that, for a long time, but it's the same thing for you. If you get this vaccine, just be careful.
Yeah. Yeah. This is a liberal country, and they don't listen.
Yeah.
So Nicole, I have a question for you. So when you were looking at this plot, you said it went down, or like what?
Yeah, because
when you look at this, what do you? Because I'm looking at the two plots separating, and as a pharmacologist, I say, okay, well, there's an effect with
because
time.
they're treated at the same time. So
Yes.
over time they separate, and the blue You see how if you look at, I look at control, and then I look at treatment. Control is red, and then treatment. So that's down. That's
Yes.
significantly reduced.
Yes.
It may not be significantly reduced. I can't see the numbers, but it starts to converge or starts to change and separate around 14 days. That's when it starts to separate. 14 days. Then you got 21. Was that 28? Yeah. So at a month, like, well, not even a month, like, what was that? 21 days is significantly reduced from what I can see. Yeah, 14. That's not significant.
Yeah.
It's down. Like if you compare red at each day, that's counted for. Each week, red, and then for day 28, and then blue, red is up, blue is down. Red is up, blue is down. That's how I look at it.
Okay.
But if you just look at blue by itself, yeah, blue just stays down. There's no change. There's no significant change. But that's after one dose.
Okay.
Because they said after one dose. My issue is the y-axis, but I have to figure out what that means. It may be like a log-based presentation, the way they presented the data on the y-axis.
Okay. Did you ever? The biggest question is why 2 doses? Did you ever explain that or had a theory on that? Why 2 doses instead of just one?
I don't know. Like we've been talking. I've been talking to my colleagues about that. And we were like, why they give 2 doses? Because that's not something that's not standard. Well, this vaccine ain't standard, but
well, that's true. Do you think that the vaccine may be borderline toxic to where they can't give it in one dose? They have to give the body time to recover before they give the second dose.
That could be that is a possibility. And now, because now that I'm thinking about how much they gave people in the first dose,
yeah,
that's a lot. Like
that's a lot. Okay.
It's like 30 micrograms.
Okay, that's considered a large dose for
30 micrograms and 300 microliters. Yeah, it's like, I mean, yeah, because it's mRNA, so it's like, it's automatically you shouldn't have to have that much. Okay, if you give somebody 30 micrograms for 300 microliters and the first dose, what is the second dose going to be? Because I was looking for that. What would the second dose be? And I know they're ongoing right now. Would it be 100 micrograms? Like what is that? Because the initial trials they were doing on clinicaltrials.gov, they were talking about, they had people grouped like 20 microgram, 50, 100, stuff like that. And they had different age groups. They had them placebo matched to different age groups. That's what they were doing. And those concentrations should show a trend where the higher the dose, the lower the infection post injection. And so that was the thing. And the other thing is the study that, I don't know if they said it in here, but they actually had to give them, like, these are healthy people. They had to give them the virus. First of all, nobody in the wild gets injected. We ain't going around injecting each other.
Okay.
You have to give them the virus. They didn't just come off the street, which would make more sense. I'm just saying. If you already sick and you probably ain't gonna make it because you're in ICU.
Yeah.
We'll enumerate your family and we will give you this vaccine and see what's going on, because your next step is hospice probably if you don't recover. You've been on a ventilator and whatnot. So you take healthy people, inject them with the virus, and then turn around and wait for them to have, some kind of response that you can determine through the blood, antibody response, things like that, antigen responses. At a certain threshold, then, okay, placebo or vaccine. That don't make no sense to me. I'm sorry. I was like, what? Huh? Like, I had that look like, what the heck? So that already kind of threw me off. But the data they have here, if you just look at this graph, it worked. It worked.
Okay.
But the methods prior to this is like, it, to me, and a lot of us that are talking about it, we were just like, this is very questionable. Like, why? But hey, it's America.
That is
This
true.
is America. I was like, you took all these healthy people, they got the vaccine, and then they had, all you were talking about, all the responses they were having as well on all those tables. I was like, my God, but they were healthy before, and you don't even know if you well, the thing that was good, I would say one thing that's good, one thing that's positive about doing it that way is the vaccine you gave matched the virus you gave.
Mm-hmm.
Well, congratulations. That's, they did that right. They gave the virus, waited, and the vaccine they gave after that forced infection, synthetic infection, was matched to that viral sequence. So that made sense, which is probably why their data is so good right here.
Mm-hmm.
It's not in the
Okay.
wild. It's controlled.
Yeah, Nicole,
I just want to see. I want to see wild experiment. Wild people on the street or something.
Well, we're gonna see it in the next 3 months.
Yeah.
So
Wow.
it's probably like a scientist's dream, boy. They'll say, "Oh, we're gonna have a big test, like 20 million people all at once, and see what happens."
Yeah. So, Nicole,
I take back what I said. I take back.
You're gonna see it in the wild, wilder than you probably want.
Dang.
So, Nicole, I was going to say that I've never, my background isn't molecular biology, but I didn't know that it was possible to inject an mRNA strand into another organism, and then it'll start just replicating it or transcribing it. I'm sorry, translating it. I didn't know that could be done.
Yeah, you can blame the God of your own understanding for that.
Well, that's what a virus.
Blame the God of your own understanding.
Basically, that's what the COVID is. Basically,
right.
Well, it does, but it has its own machinery.
Yeah.
Not much, but it has the ability to hijack your machinery.
Yeah.
That's what viruses do. They're terrorists. They're little hijackers. They ain't got their own stuff. You have to. They don't come with any tools.
Yeah, that's
show up. Hey, and then I'm gonna take all this and this, and they just use your stuff.
Yeah,
it hits the mutations you get later on.
That's what concerns me. Ordinarily, what a vaccine has a dead virus. It's not active. You'd inject it, but it has the characteristics to actually cause an immune response. Okay,
right.
But this is not a dead virus. It's supposed to replicate, which bothers me. I said, okay, what is the short-term effect? Would be fine. It might do what it's supposed to do. The thing is, like I said, my question is, does it completely terminate? Does the body completely eject the stuff? Does the body completely basically get rid of all these little creatures, or do they just keep floating around and keep doing this? Or,
the shedding is a possibility, but that, like I said in the chat earlier, that's based on your immune system. If you are immunocompromised, you're pregnant, you're an older person, you got a lot of pre-existing conditions, then the shedding of that is how effective will that occur? Like how quickly will that occur for you? If you are dealing with if you're already they give you the vaccine and you're already sick, but you don't know you're sick. Are you expected to shed the virus that you had before the vaccine and the particles that were in the vaccine like that? I'm like, what? How does that work?
Yeah,
because are they testing you before they give you the vaccine?
I hope so.
That's the thing. Everybody on the news, they getting the vaccine. I'm like, wait, and then we're watching the news, and I was like, hold up, Sanjay Gupta. Hold on now. Did y'all test these people before you brought them on CNN to be in the camera and stuff? Do they have a test that's negative? Because if they got a positive test, y'all dumb as hell. Straight up, you're stupid. What are you doing? You're giving them double infection. You're transfecting them twice. What are you doing? You don't do that. You're mixing, even if it's the same sequence, which is probably not possible. If you got infected in February and then you recovered, there's antibodies for that strain.
Right. Yeah.
And then you get the vaccine in December. Your immune system is like, we've seen this type of thing before, but we're not sure.
Right.
Or your immune system will say, get them. And then there's a huge cytokine storm and you're dead in 2 weeks.
Yeah.
That I worry. I'm very concerned with that. Like that's the worry for me, because looking at the molecular biology of it, like you're saying, Dr. Dunbar, that the machinery is there. It's already there. It's primed and ready to go. And the cells are so naive to this new foreign particle that's there that they will stop what they're doing to translate that and get it out the way and then go back to what they're doing. It's in the muscle cells. Muscle cells are constantly, constantly going through translation. Like I said, we use our muscles and you're putting it into your shoulder and your arm or whatnot. And that translation happens so quickly, with this type of, it's a nucleoside capsid molecule that they have with messenger RNA. It is not dead. It is alive, which is why it has to be frozen, because what Don Calypso and I were talking about, you have to keep it, because somebody said, like, why is this store so cold? Because mRNA it's dead if you put it at room temperature. You can't do anything with it. I've done it by accident before, leaving it in the bucket overnight, of ice, and I left some samples in there. The next day they were floating in the water. I was like, oh damn. So I tried to use it. It was nothing. It just, I compared them to real samples that were not left out, and it didn't work. DNA, you can leave at room temperature, you can leave it 4 degrees, no problem. RNA, it just falls apart.
It's too fragile.
It's single-stranded. Even if it's double-stranded, there's some single-stranded parts, and it's naked. mRNA. There's nothing there holding it together, keeping it, safe, for lack of a better word.
So you had to put it in like suspended animation by freezing it.
-80.
Yeah, that's When that kind of bothered me when they had to go like to -80. I said that's pretty damn cold.
Yeah, well, I use it every day. Yeah.
Oh, you do? Okay, well, see, that's why we have people. That's why we have people like you.
That's why every time I make RNA, or not make RNA, but every time I isolate RNA from my cells or tissues or whatever, I quickly reverse transcribe it into cDNA, and then the RNA can just stay. It's total RNA that I'm isolating, so it's all the RNAs in the cell. But only mRNA can be reverse transcribed to make cDNA, complementary DNA. So it's the mirror image of the parent strand that RNA came from. If that makes sense, because I don't have DNA. So all I have is RNA.
So we got to store our vaccines next to our quantum computers, huh?
I don't know. But -80 is standard. -95 that's not standard in laboratories.
Wow.
But
Yeah, like I said, this is I'm glad we have you guys here, because this is just bizarre. Like when I would have never even thought about it, but being around you when they say mRNA, I said, okay, this is different.
I'm going to show a little cartoon here so people can kind of digest what Nicole was talking about regarding the mutation, but also what a traditional vaccine, how a traditional vaccine will work. So normally the patient would get injected with the weakened virus, and they would build the antibodies up against it. But what Nicole was referring to in terms of the virus mutating, if you look at the bottom there, if you look at the top, those antibodies were against those spikes, but that virus mutated so that that antibody cannot recognize those new spikes. That's what she was referring to.
Just bounces off and goes on its way and dies. The antibody just dies. And that's another thing. Like you can have, the immune system, the T cells, B cells, they know something's there that shouldn't be there, but there's no antibodies. Like they can't send anything out there to capture this thing and try to learn what it is. So like that, when they said mRNA, I was like, oh, this is bullshit. Like I was like, this is not going. I was like, this is.
You think it's a flawed strategy?
Yes, yeah, I really do, because
interesting.
I think about like I was like, man, I was going through my notes, looking through like, when is the last time I put mRNA? I ain't never put mRNA in nothing. Like I've taken mRNA out. You know, I just sent RNA. What was that? New Year's Eve. The day before New Year's Eve, we sent mRNA to be total RNA. Excuse me, total RNA to be sequenced. And they had to go through the quality control and everything to make sure it is total RNA. And they tested everything. And I asked them. I said, "Can you isolate an mRNA and send me back an mRNA sample? I want to look at some specific sequences for myself." Y'all already know what it is, but I want to look at some specific sequences for myself, of this virus, and see if I could match. I'm trying to like match and see like, okay, if I were to do something, injection or something or transfection, how would I go about that with mRNA? How could I do that? It's so hard to do that type of work. Like you have to be in a cold room, literally a room that's 4 degrees, doing this type of work.
Wow. Okay.
You can't be in a standard BSL-1 or BSL-2. You have to like a BSL-4, of course, doing this type of work in a cold room with a bunny suit on, with the tube coming out your back, coming out the back of the suit with the hood on, because everything has to be so cold and you can't do but so much a day. Like I looked at it, I said, there's no way, like how you, no, mRNA is not, that's just, it's like, it's not, and see the way they have this mRNA is prepped and primed for translation. It's not like it needs any processing or anything like that. It's just ready to go.
No, okay.
Like the picture you have.
So as soon as you inject it, it starts working.
That's why, that's why people having all these effects like so quickly.
Okay.
In the muscles and in the Bell's palsy, that's a muscular disorder of the face, the facial structure. And this is not only it's not only in your muscles, it's in your bloodstream.
Okay.
So it's systemic, which is what you want. You want to have a systemic effect, but 15 minutes one nurse, I forget, it was a Black lady. She's a nurse. I forget where she is. But her effect came, her response, her Bell's palsy came like 3 days later. And she said it was not gradual. It's just she woke up and her face was twisted like she had a stroke.
Wow. Okay.
She was like, what is going on here? And she's like, I can't smile. She said, I'm trying to smile right now, but I can't. Her brain is not able to talk to her facial muscles and tell them to, go up into a smile. And so that I was like, this is some what? Now it will subside, but the fact that you have it, if you are, for instance, if you're pregnant, then and you have Bell's palsy, you don't know what effect Bell's palsy has on the fetus, as on your child. That's my issue. Like, and people who are older, if you are on certain medications, how does that Bell's palsy affect, contradict your medications? How does that, like, does it make it worse? Or, if it's a blood pressure medication or something like that? That's what I worry about for my grandmother, for instance. But I'm gonna see what Judge, he said. I'm not trying to scare you, Judge. I'm sorry.
Okay.
Yes, in a nutshell, yeah, I've been saying it. Something is fishy.
When the CEO won't take it, then something's fishy. Then
what? That was like,
well, I think the interesting thing.
Yeah, when the guy that owns the company won't inject himself with his own
I was
product,
like, this fool here.
he knows something.
Yeah.
Well, I think the interesting thing is that it's a 2-year study, so it's not going to wrap up until what, 2022?
It won't. Yeah,
We
it won't
won't know.
up. Yeah, you're right. 2022.
The extent of what the vaccine will do.
I don't know if they're oh, that's true. The long-term effects.
Yeah.
And that's just I'm thinking something like, say, like a chicken pox, right? That can embed itself into your nerve sheets, your and stay there, dormant for years. Like I said, this is it does it worries me. It doesn't scare me, but it worries me because this is like cutting edge, and putting something cutting edge into 8 billion people's arms is like, kind of worries me. you'll find out quickly, at least the short-term effects, what it does and what it's not going to do, because you have a big enough sample. But my thing is the long-term effects. You know, like I said, it does your body shed this stuff? Because it's not one of those things where you have a whole dead virus where it's not going to really be active. It's not going to do anything, just cause an immune response. But this is actually active. Actually active and and it will replicate itself, which does bother me.
Mm-hmm.
That I really it really bothers me. And then the fact that it's just, the main thing is they just rushed it. So when you look at safety, I'm like, how can you be safe when you rush this thing?
Yeah, well,
I
that's good.
understand, they're trying to get the economy back on and everything. But it's difference between rushing a standard, traditional vaccine versus rushing a vaccine that nobody's ever taken before.
Yeah, I'm sure it's making Dr.
That was
Dunbar's head spin.
No. I mean, it just is what it is,
Yeah.
As you always say. Yeah. Well, Nicole and BGS is almost 11 o'clock here on the East Coast.
All right, sir. I appreciate you guys. Hey, you guys, if you haven't hit the Cash App, man, because you're getting this stuff raw, and because I think anybody else probably will give it to you filtered. So if you appreciate his work, we appreciate Nicole's work. Nicole should put her Cash App in, because experts are expensive, and you got two of the best. I do mean it, two of the best. I know them, and Nicole is brilliant. And
I think one of the cool things is when Nicole came in, she said, "Okay, Dr. Dunbar, you are approaching this as a toxicologist and a pharmacologist, but I'm looking at this a different way." Yeah. That's what happens with this stuff.
Well, basically both ends, because she's a creator and you're an evaluator. Yes. So you're getting both ends of the spectrum. So that's good. Because Dr. Dunbar is worried about what are the after effects once, possible after effects that could happen once it gets out into the wild, once it gets out to the public. And basically Nicole is worried about how is this thing designed. So you're getting both ends of the spectrum. So this is highly unusual, okay? Especially in a space like this. So, hit Dr. Dunbar's Cash App. Please subscribe to his channel, because this is probably going to be ongoing. Make sure you subscribe to Nicole's channel. Because trust and believe, you two have changed a lot of my friends' minds, because they were staunchly against this, and they thought it was fake. And you guys personally changed their minds. I said, okay, if you don't believe me, because I don't know anything, believe the people that actually do this for a living. And so I appreciate you.
Okay. Well, Master Teacher, you've been banging the drum all along. Some folks have been like, it's a hoax, it's not real. But you've been the main one saying, okay, look at New York, look at the bodies, look at this, look at that.
Well, you know what? My thing is, after, because I've been following this kind of stuff for like 30 years. And I know something's up when people are trying to hide something. And when the Chinese were trying to hide it, I said, this is real. This is not a hoax, because they're too busy trying to suppress this information. As I said, this thing must have legs. I didn't know it would be this big. And when Nicole says this is a real thing, and this probably could have been worse, I said, all right, this is happening. The thing is that now, how fast will it mutate? How long will it stay around? Because the only good thing about basically the H1N1, the original H1N1, which is the Spanish flu, it mutated to be benign or less lethal.
Right.
Will this one follow the same path, or will we be dealing with this for the next 100 years? We don't know. That's the problem when these things get out.
Mutate down, or mutate to a, like you said, a benign, less or more. It's more indolent, just benign, less aggressive. But this one's mutating up, as it's supposed to do. It's designed to do that. And so, if I design something like that, I would have it to mutate up that way. Honestly, like that's what I would do, because I want to make sure, like, what we have, as far as the technologies that we can access, the cells they adapt, and they kick out what you put in. You're forcing genetic material into them, and they're like, after so many generations, they kick it out. You go and test them, and they don't have the expression anymore. So if I were doing something, I would make sure that no matter what that cell did to adapt, whatever I infected them with, it would consistently, not only, it wouldn't just be infecting them, it would consistently express over and over and over. And so like, I don't have issues with that. My issue is like with the if you're going to do something that you say is 95% effective, it better freaking work. And, efficacy comes after safety. When you do trials, like Dr. Dunbar knows, you have to have safety first, then your efficacy. Don't go on TV saying it's 95% effective and your CEO don't want to take it.
Yes.
Because then I'm gonna call you on some BS on that. Like, that don't make no sense. But yeah, I knew it was real. My mom was calling me way back, and BGS, we were talking about this and everything way back earlier last year. And my mom was like, "Oh, well, this is just a regular flu, regular flu." I was like, "No, it's not." I dang near cussed my mom out. I said, "Are you kidding me?" Like, I was like, "What?" I was yelling at her, and she was like, "What you yelling at me for?" I was like, "Mama," I said, "you do not need." I said, "Matter of fact, just stay in the house." Okay, just stay in the house, because don't go outside. Just stay at home. Like, this is before they said anything. I was like, "Look, you don't know what you're talking about. You don't know what you're messing with. You really just need to like listen to me."
Yeah.
I'm not playing around. Like, we don't be having meetings every day. We don't have meetings like that all the time, about something like this. And they got the pictures on the screen and, showing all the sequences and all this stuff. They don't normally do that. And then they all of a sudden did that. And I was like, what? So I told her, I said, "Ma, you need to be careful." I mean, I said, don't go nowhere. That was early. I told Jason, I told his family, I was like, "Please be careful." I said, "This is going to get worse." That was like February. I was like, "It's going to get worse. Just be careful. Like, if things start shutting down, this is why, and in March, things are shut down." I was like, well, I looked at the sequences and the structure ahead of time, and I was like, okay, this is not regular. Like, this is not standard sequence. There's a lot of we call them pockets, a lot of little pockets of sequences that don't make no sense, that just don't do anything. And I'm like, well, they don't do anything because when you compare those sequences to standard sequences on NCBI, there's no match. Yeah. So there's no function, but we didn't know the functions at the time. Now they know the functions, and they're related to these mutations that we're seeing. So at that time, I knew that this was not like, the flu virus. It was not like, and people kept saying, "Oh, this is a regular virus." I'm like, man, you gonna die. Like, you gonna die. Like, you don't know. Like, this is hopefully everything is a learning experience for everybody. Like, don't wait for the media to tell you what's going on. Like, just don't, listen to people who actually do the work.
Yeah.
Regulatory people like Dr. Dunbar, very important, because they know like a lot of cutting edge stuff. They know stuff before I know it. I'm the one who has to do the work and go in the lab. But a lot of regulatory people, they know way before we know. In advance. They're the ones who have to like make sure that we're doing what we're supposed to be doing. That's right. And I know, like, and especially from where Dr. Dunbar is, yes, a lot of those people that's in DC, they know what's going on way before in Atlanta. They know before we know. I'm on the ground level. Super Tree is the type of person like him, Michael. We're on the ground level of stuff, face to face with people. We're not the higher echelons of regulatory and things like that. But I hope everybody just stays safe. That's my main thing. Please just don't take your life for granted. Please don't do that. That's all I can say.
Yeah, it's stay tuned, and I think you and Dr. Dunbar said it from the very beginning. Don't just run out and take this. Wait and see what happens.
True.
Yeah. Well, Nicole, I appreciate you coming through and letting us know what you know, and I look forward to hearing some more of what you have to say about this thing as it continues to unfold.
Sure. Thank you. Thank you for having me up here.
Okay. And BGS, as always, thank you.
All right, Dr. Goodbar. inside joke. You had to been on the other stream, talking about talk about swag, and then you'll understand the joke.
It's just funny when you said it. Like I have to go back and but that's just
It,
funny
yeah,
when you
yeah,
said that.
it's an inside joke. I know he would laugh when I said it.
But I'll talk to y'all later. Y'all have a good evening.
All
You too,
right,
Nicole.
peace. And
Peace.
thanks, Dr. Dunbar.
Thank you, BGS. Okay, everyone, I'm going to read the safety section from this memorandum, and then I'm going to stop. This is lengthy, but I think it's a good way to finish this up. So let me just enable my screen share. And again, this is below this video in case you guys want to read it for yourselves. It's a long document, but this is the safety summary. So this reads, the information provided by the sponsor was adequate for review and to make conclusions about the safety of the vaccine in the context of the proposed indication and population for intended use under the emergency use authorization. The number of participants in the phase 2/3 safety population meets the expectations described in FDA's guidance on development and licensure of vaccines to prevent COVID-19 for efficacy, and the median duration of at least 2 months follow-up after completion of the 2-dose primary vaccination series meets the agency's expectations described in FDA's guidance on its emergency use authorization for vaccines to prevent COVID-19. The all-enrolled population contained more participants greater than 16 years of age, regardless of duration of follow-up. The demographic and baseline characteristics of the all-enrolled population and the safety population were similar, although the overall median duration of follow-up in the all-enrolled population was less than 2 months because the protocol was amended to include subpopulations such as individuals with HIV and adolescents. The data from both populations altogether provide a comprehensive summary of safety. Local site reactions and systemic solicited events after vaccination were frequently and mostly mild to moderate. The most common solicited adverse reactions were injection site reactions, fatigue, headache, muscle pain, chills, joint pain, fever. Severe adverse reactions occurred in 0% to 4.6% of participants, were more frequent after dose 2 than after dose 1, and were generally less frequent in adults greater than or equal to 55 years of age as compared to younger participants. Among adverse events of special interest, which could be possibly related to the vaccine, lymphadenopathy was reported in 64 participants, 54 in the younger age group, 10 in the older age group, and 6 in the placebo group. The average duration of these events was approximately 10 days and 11 events ongoing at the time of the data cutoff. Bell's palsy was reported by 4 vaccine participants. From dose 1 through 1 month after dose 2, there were 3 reports of Bell's palsy in the vaccine group and none in the placebo group. This observed frequency of reported Bell's palsy is consistent with the expected background rate in the general population. There were no other notable patterns or numerical imbalances between treatment groups for specific categories of non-serious adverse events that would suggest a causal relationship to the vaccine. Okay, a little bit more. A total of 6 deaths occurred in the reporting period. In the vaccine group, 1 participant with baseline obesity and pre-existing atherosclerosis died 3 days after dose 1. And the other participant experienced cardiac arrest 60 days after dose 2 and died 3 days later. Of the 4 deaths in the placebo arm, the cause was unknown for 2 of them, and the other 2 participants died from hemorrhagic stroke and myocardial infarction, respectively. 3 deaths occurred in the older group. All deaths represent events that occur in the general population of the age groups where they occurred at a similar rate. The frequency of non-fatal serious adverse events was low without meaningful imbalances between study arms. The most common systemic adverse events, I believe that's systemic, in the vaccine arm, which were numerically higher than in the placebo group, were appendicitis, acute myocardial infarction, and cerebrovascular accident. And in the placebo arm, numerically higher than in the vaccine arm, were pneumonia, atrial fibrillation with syncope. Appendicitis was the most common SAE. In the vaccine arm, there were 12 participants with SAEs of appendicitis, 8 in the vaccine group. Of the 8 appendicitis cases in the vaccine group, 6 occurred in the younger age group and 2 occurred in the older age group. One of the 6 participants with appendicitis in the younger age group also had a peritoneal abscess. Causes of appendicitis in the vaccine group were not more frequent than expected in the general population. While not observed in clinical trials, 2 anaphylactic reactions in the immediate post-vaccination period have occurred with the use of the vaccine in the United Kingdom in individuals reported to have prior history of anaphylactic reaction. The components of the vaccine that may have triggered these anaphylactic reactions are unknown at this time, and the 2 individuals were not reported to have known history of allergy to specific components of the vaccine. So I'm going to stop that there. But before I cut this off, let me just make sure I gave you guys the right definition of SAE, because with science, there are a lot of terms. So SAE, serious adverse event. So it's not systemic adverse event. It's a serious adverse event. All right. Everyone, thank you for watching this live stream. Thank you for Nicole and the master teacher coming up and participating in this stream and asking questions and adding to the discussion. Thank you all for watching. Thank you to all of those of you who donated. Look out for more content on this. And I just want to reiterate that I didn't do this to necessarily convince someone to take the vaccine, but just to put more information out there from a scientist's perspective, and also to give my point of view on what's known from a scientific perspective. And also to, because when something like Bell's palsy, when someone goes on Twitter and says they developed Bell's palsy after taking the vaccine, unless you know that subsides, in most cases, or in many instances, after a short period of time, you could go on thinking, okay, well, once I developed Bell's palsy, I'll have it for the rest of my life. So with that, I'm going to stop. Please let me know if you have any questions or comments in the comment section below, especially if you're watching this on the playback. And if you go to your primary care physician to get the vaccine, make sure you ask questions. And those memorandums are in the description box below this video, so you guys can read that stuff for yourself. So yeah, so everyone be safe out there. Please like, share, and subscribe. And as always, remember that your attitude determines your altitude. Take care, and I'll talk to you the next time. Bye-bye.